Hi guys, this is Dr. Momsilovich with your lecture 10.1 on population characteristics. So if you think back to one of our first lectures, we talked about the fact that there's different levels of biological organization. There are cells and then tissues and then organs and organ systems and then individuals. Up to this point, we've been talking a lot about individuals and individual differences. We're now going to be talking about populations, which is the next level up. So a population is a group of individuals of the same species living in the same area at the same time. When we're talking about population characteristics, we sometimes have to go backwards and look at the characteristics of individuals because sometimes those characteristics are still important. And a lot of the same rules on individuals still apply when we're talking about populations. So let's first start by looking at a few parameters that we need to discuss when we're talking about populations. So first, remember that a population is a group of organisms of the same species in a particular area. Now think back and remember that a species is members of a group that can interbreed and produce fertile offspring. And your area is also going to be important because we can discuss the number of people in the city of Phoenix, the number of people in the city of Arizona. We can discuss the number of people in the United States of America. And so your study area is also important. And so once we know those things, the species that we're looking at, and our area, we can start asking other questions such as how many individuals, and we denote the number of individuals by the number n, or sorry, the letter n, not the number n. We also look at how they're spread out within that area, so their density and distribution, and we'll talk about different patterns as well as how dense they are. We also need to look at the age and the sex of individuals. So there are different age structures. Is your population made of a lot of young individuals or older individuals? Is there an equal sex ratio? So an equal number of males and females, things like that. And that also relates to your growth rate, which is what determines how many individuals there will be in the future. So first we need to discuss how we can count individuals in a population. And we actually use very similar methods to the methods that we use to count the number of people in the country or on the planet. So for example, this year is a census year in the United States, which means that the government is trying to get an idea of our population. And so a census is where you literally just directly count the number of individuals in your population. Another way to determine how many individuals are in your population is through an estimate. And an estimate is often easier, but it can lead to some misleading results where you either overestimate or underestimate your individuals. So for example, in animals, we can count nests or subpopulations using transects and traps. But this would be similar to, for instance, counting the number of houses in the United States of America and then assuming that there's an average number of people in each house. We would probably end up underestimating the number of people if we assume that there's only 4 people, as we assume a standard family of 4 in every house in the country. And so estimates can be useful, but they're not going to give us as exact of an answer as a census. So the number of individuals is represented by the symbol N. So for example, N is equal to 21,077 southern white rhinos on the planet in 2015. So that is the population size of white rhinos on the planet in that year. One method that scientists use often to estimate the number of individuals of a species in an area is called quadrats. This is where you count all of the individuals in a designated area within your study area. These are traditionally set up as a square, so quad means four, so it's all species that occur within the four walls of this square. So you would place or you would mark within your study area different locations for your quadrat and then count the number of individuals within each quadrat. The size of your quadrat would depend on the species. So for example, if you were looking at bacteria, you would want to do something small, like 1 centimeter squared. For small plants, you would do something like 1 meter squared quadrats. For something larger and animals that can move around a bit, 10 meters squared. For medium-sized trees or small mammals, 100 meters squared. And for large species, you would do something more like 1 kilometer squared. So when you're setting up quadrats, you will generally choose your locations for the different quadrats randomly within your study area, so that once you get there, you're not tempted to put your quadrat where you do see your species of interest already. So you, ahead of time, choose where you're going to place your quadrats, and you want to place 20 or more. Statistically, 30 to 50 is actually the best. So you would choose quadrats within your test area and then extrapolate the data to determine your population size. So for example, in an area of 1,000 kilometers squared, we might want to find the population of oak trees. So we would place quadrats within that study area so that the total number of trees that were counted in 20 of the kilometer squared quadrats, and so say that we have 750 trees in 20 kilometers squared. We then extrapolate the data so that we find that there are 750 trees in 2% of the area, because it's 20 out of 1,000 kilometers squared. So therefore, in the whole area, we would expect there to be 37,500 trees. And so using this, we can estimate the number of individuals of a population in a given area. You can also use satellite imagery to look at the number of individuals within a designated area. So if we're looking at oak trees, if you know roughly how big the canopy of an oak tree is, you can use satellite imagery to then confirm or back up your quadrat sample. Quadrats are useful when we are looking at the number of plants or other things that don't move around very much. For animals and things that do move around, scientists often use mark recapture methods to estimate the number of individuals in a population. So this is where you would set traps and then capture individuals of the species of interest. And then you mark them in some way and release them back out into the wild. At a later point, you're going to set traps again and capture them. And then you're going to look at the ratio of individuals that you catch that were previously marked compared to individuals that you did not already catch. So you look at the ratio of previously marked to unmarked individuals. And then those numbers are used to estimate N, or your population size, using a very simple equation, which is that the number of marked first catch individuals times the total number of second catch divided by the number of marked second catch. And that equation will give you your population size. When scientists are marking different animals, they have different methods for doing this. So on the bottom left-hand side of your screen, you have a species of ants that's actually studied at ASU. And these are really, really tiny ants. They're called house hunter ants. And they're so small that they can actually fit in between slides of a microscope. And so the scientists who are studying these ants will do different marking color patterns on them to do mark recapture, as well as AI simulations where they can have a computer track the movements of a single ant to see how different individuals are moving within a colony. And so those scientists will do different patterns of colors and markings on the head, thorax, and abdomen of each ant to be able to determine different individuals from each other. For something like birds, scientists will actually do bands on the wrists of the birds, and so there will be color markings where you'll have 2 or so on one and 1 or 2 on the other, and the pattern and colors will determine the individual, and then you can track different individuals within your population. For other species that migrate a lot, you will end up or what scientists do is they use tags that are able to track different information about those species. I did a bit of fish research during my PhD in Utah, and we did some mark recapture on fish. And so as you can imagine, you can't really put a band or anything that sticks to a fish or paint on it because it's going to wash off in the water. And so what we did was we actually took a needle and essentially did a tattoo on the fish where you inject ink underneath the fish's skin in certain locations, either near the gills or the tail, and on either the right or left side. And that's how we were able to determine the each individual within our mark recapture. Next, we have to talk about density and distribution. So density is just the number of individuals per unit area. So that is going to depend on, again, the size of your study area. So you could ask how many people occur within each acre within a city, within each hectare, or you could look at the number of individuals within a square mile. And so that number is going to be different depending on your study area. And it's also going to depend on the space that you're looking at. So for people in particular, you would expect fewer people per square mile in a rural area or a smaller city like Phoenix compared to a mega city like New York City or Hong Kong or Tokyo. We can also look at the distribution of organisms within a study area. So there's different patterns of how different organisms distribute themselves within a certain area. So uniform distribution would occur in something where there's an equal amount of distance between each member of the species. So this would occur generally in things that are territorial. So not necessarily penguins, but other birds generally have a territory and they keep other individuals of their species out of their territory. And so they're sort of uniformly distributed within their environment. Random distribution would be where the space between one member of a species and another member of the species is just random within their environment. So it's not based on competition or anything like that. It's just sort of randomly determined where individuals are found within that environment. The last type is called clumped, and this type of distribution is seen in animals or organisms in general that occur in herds or groups. And you would also see it if your resources are not evenly distributed, if they're concentrated into one area. So something like water. If there's only a certain amount of water in an area, then organisms might be clumped around that resource instead of being evenly distributed throughout their environment. When we're looking at population structure of a population, we use what are called population pyramids to give us more information about the demographics of that population. So within that, we can see the number of individuals that occur in each age category, and that gives us information on birth rates and death rates, as well as the number of individuals that are that survive after birth, and whether there's a difference between males and females that survive in your population. So this can show us how many reproductive aged individuals there are. So it can also show us, are there survival differences between males and females? And are there signals of previous challenges in your population, something like a drought, which would be shown by having an indent into your population. And so the population structure of your population will determine the shape of this pyramid and whether it looks like a pyramid or if it looks a little bit different. So we're going to go through a couple different examples. So here we have two different population pyramids, one for Afghanistan and one for Canada. And you can see in each of them the number of males versus the number of females. And you can also see how many individuals there are of different ages or life stages. So if you look at Afghanistan, there are about 2.4 million individuals for both males and females that are between the 0 to 4 age range. In Canada, there is only about 0.8 million, which is about 800,000 individuals in the 0 to 4 age range for both males and females. If you look up a little bit higher on that pyramid, you can see that in Canada, more of the population occurs in the age range from about 40 to 60, whereas in Afghanistan, more of your population is in that younger age group between 0 to about 40 years of age. And so that's important because in Afghanistan, what it's showing is that they have a younger population and that their population size is growing because you have individuals that are in that reproductive range. So usually we would say about 15 to about 40 years old would be typical reproductive age. And you also have a lot of younger, or a lot of children younger than that who in the next 5 to 10 to 15 years will enter that range of reproduction and then also be able to add to the population. In Canada, you have more individuals in the older age category that are not reproducing and fewer in that reproductive category. It also looks from this pyramid for Canada like the individuals that are reproducing are having fewer children than the individuals in Afghanistan. And so because of that, the population size of Canada is actually shrinking while the population size of Afghanistan is growing. And so we can see also from this the death rate where individuals in Canada are living longer lives than individuals in Afghanistan because there's more individuals in those upper age categories in Canada than in the Afghanistan. So the last slide, I'd like you guys to go back and watch for yourselves. It's a video on some population counting techniques. You're going to be using a different type of population estimating technique in your lab for week 10, but I do want you guys to go back and get some more information on this by watching this video.